Hi everyone, welcome to all. This is Vidyam Academy's NEET Initiative Most Expected Question Series. This is Rajiv Patel. Today's topic is 12th Biology, second chapter, Human Reproduction. Into this subtopic is the male reproductive system. First question is concept-based question. The temperature inside the scrotum is approximately 2 to 3 degrees centigrade lower than the core body temperature. Which of the following best explains why this condition is essential for the normal spermatogenesis? Option A, higher temperature enhances testosterone synthesis. Option B, enzymes involved in spermatogenesis function optimally below core body temperature. Option C, blood flow to testes requires cooler surroundings. Option D, Leydig cells cannot function at body temperature. What is the answer? Would you guess? Yes, the answer is B. Why and how? Let's see the explanation. Spermatogenesis requires a temperature 2 to 3 degrees centigrade below core body temperature. Because the enzymes catalyzing sperm cell division are heat sensitive, the scrotum acts as a thermoregulatory organ to maintain this optimal microenvironment. That's what option B, which I have given here, is the right answer, what exactly the first question demands for. Next, let's move to second one. It's related to structure. A student lists the correct sequence of ducts through which sperm travel from the testis to the outside. Which sequence is accurately matched here? The first one, A, rete testis, efferent ductules, epididymis, vas deferens, ejaculatory duct, and urethra. So this is the first sequence, option A. Option B, seminiferous tubules, rete testis, vasa efferentia, epididymis, vasa deferens, ejaculatory duct, and urethra. So this is option B. Option C is seminiferous tubules, epididymis, rete testis, vas deferens, urethra. Option D, rete testis, epididymis, vasa efferentia, ejaculatory duct, and urethra. Your time starts now. Could you guess? Could you guess the answer for this question? Yes, the answer is B. The correct pathway is seminiferous tubules, rete testis, vasa efferentia, epididymis, vasa deferens, ejaculatory duct, finally urethra. So this is exactly correct sequence which I have given under below the option. Option B rightly matched, and it's a proper sequence what exactly question demands for. Question number 3. This is very important question, assertion and reason based question. So choose the correct option. Assertion A, Sertoli cells are called nurse cells of the testes. So here the reason is or Sertoli cells provide nutrition and structural support to developing spermatogenic cells and secrete inhibin to regulate FSH secretion. So here the options are option A, both A and R are true and R is the correct explanation of A. Option B, both A and R are true but R is not the correct explanation of A. And C, A is true but R is false. Option D, A is false but R is true. Once again, Sertoli cells are called nurse cells of the testes. Sertoli cells provide nutrition and structural support to developing spermatogenic cells and the secrete inhibin to regulate FSH secretion. What is the answer? Your time starts now. Yes, the answer is, anyone? Would you guess? The answer is option A. Sertoli cells nurture developing germ cells. We are calling it as a nurse cells by supplying nutrients and essential proteins. So this is how we can see so that Sertoli cells are called nurse cells of the testes. They also secrete inhibin, which provides negative feedback on FSH, making R is a complete and a correct explanation of A. That's what. So here both A and R are true and R is the correct explanation of A. And next move to question number 4. It's also very important model. It's an application based question. Question number 4. A 28-year-old male undergoes vasectomy, surgical cutting of vas deferens. Which of the following statements correctly predicts the post-operative outcome? Option A. Testosterone secretion will decrease significantly. Option B. Spermatogenesis will completely stop. Option C. Spermatogenesis continues but sperm cannot be ejaculated. Option D. FSH and LH levels will drop to 0. What is the answer? Answer is, would you guess? Answer is option C. Why and how? Let's see the explanation. Vasectomy severs the vas deferens, blocking sperm transport. Spermatogenesis in the testes and testosterone secretion by Leydig cells continue normally, since the blood supply and the hormonal axis remain intact. The male remains fertile at the cellular level but functionally infertile. That's what. Spermatogenesis continues but sperm cannot be ejaculated. This is the answer of the 28-year-old male undergoes vasectomy, surgical cutting of the vas deferens. The following statements correctly predicts the post-operative outcome. Next move to question number 5. It's a gland function question. Match the accessory glands of the male reproductive system with their primary secretory contribution to semen. So here the options are option A, seminal vessels, acidic pH, prostate and fructose, bulbourethral lubrication. This is option A. Option B, seminal vessels, fructose and prostaglandins, prostate and alkaline fluid, bulbourethral pre-ejaculatory lubrication. This is option B. Option C, seminal vessels, testosterone, prostate, sperm motility factors, bulbourethral, fructose. This is option C. Option D, seminal vessels, lubrication, prostate, fructose, bulbourethral and alkaline buffer. What is the answer? Match the accessory glands of the male reproductive system with their primary secretory contribution to semen. What is the answer? Option B. Seminal vessels contribute 60% of the semen volume, rich in fructose. So it is energy for sperm and prostaglandins. The prostate secretes alkaline fluid containing PSA, prostate specific antigen, to neutralize vaginal acidity. Bulbourethral glands secrete a mucus fluid before ejaculation to clean and lubricate the urethra. So this is the explanation perfectly defines and explains the male glands, reproductive system and accessory glands to the primary secretory contribution to the semen. Seminal vessels, fructose, prostaglandins and prostate, alkaline fluid, bulbourethral and pre-ejaculatory lubrication. Next move to question number 6. It is structure based question. In a cross section of the human testis, the region labeled X lies between the seminiferous tubules and the contains interstitial cells. Identify X and state its primary endocrine function. Option A, rete testis produces inhibin. Option B, Sertoli cells secrete estrogen. Option C, Leydig cells in interstitial space synthesize testosterone under LH stimulation. Option D, epidermis stores and matures sperm. What is the answer? Your time starts now. Yes, the answer is C. The interstitial space between seminiferous tubules houses Leydig cells. Under the influence of LH from the anterior pituitary, they synthesize and secrete testosterone, the primary sex male hormone essentially for spermatogenesis and secondary sexual characteristics. So that's what Leydig cells synthesize testosterone under LH stimulation. So this is the right answer, what exactly the question demand for. Next, move to question number 7. It is assertion and reason based question. Once again, we have already seen earlier the same kind of a question. So here the assertion A is the epididymis is essential for sperm maturation and storage. Reason R, sperm leaving the seminiferous tubules are immature and non-motile. They gain motility and fertilizing capacity during their transit through the epididymis. So here the assertion is A and reason is R. Option A, both A and R are true and R is the correct explanation of A. Option B, both A and R are true but R is not the correct explanation of A. And option C, A is true but R is false. Option D, A is false but R is true. What is the answer? Could you guess? What is the answer? The answer is option A. Assertion A also right and the reason R also right. Why and how? Let's see. Freshly produced spermatozoa from seminiferous tubules lack motility. Once again, freshly produced spermatozoa from seminiferous tubules lack motility. The 4 to 6 meter long epididymis provides the environment for sperm to mature, acquire forward motility, and develop the capacity to fertilize an egg, making R is a precise explanation of A. That's what here both A and R are true and R is the correct explanation of A. And next move to question number 8. It is clinical application question which most important for your examination. A male patient is diagnosed with cryptorchidism, undescended testis remaining in the abdominal cavity. The physician explained that this condition causes infertility. The most accurate biological explanation is for option A. Testosterone cannot be produced inside the abdomen. Option B. LH and FSH cannot reach the testis in the abdomen. Option C. The higher abdominal temperature inhibits the temperature sensitive process of spermatogenesis. Option D. The epididymis cannot store sperm inside the body cavity. So here the options. Would you guess the right answer? Yes, the answer is C. Why and how? Let's see the explanation. Spermatogenesis requires a temperature 2 to 3 degrees below core body temperature. In cryptorchidism, the retained testes are exposed to the full abdominal temperature, approximately 37 degrees centigrade, which denatures heat-sensitive enzymes of spermatogenesis, leading to azoospermia, no sperm production, and infertility. That's what this is exactly explains why option C is the right answer, what exactly the question number 8 demand for. Question number 9, hormonal regulation related question is this. Which of the following correctly describe the hormonal feedback loop regulating spermatogenesis in males? Option A, GnRH anterior pituitary, FSH only, Leydig cells, testosterone, negative feedback on hypothalamus. Option B, GnRH, anterior pituitary, FSH and LH, Leydig cells, testosterone and inhibin exert negative feedback on hypothalamus and pituitary. Option C, LH, Sertoli cells inhibin, Leydig cells and testosterone, positive feedback on GnRH. Option 4, GnRH, posterior pituitary, FSH and LH, Sertoli cells, testosterone. What is the right answer? Option B. The hypothalamus releases GnRH, anterior pituitary releases FSH and LH. So here FSH stimulates Sertoli cells supporting spermatogenesis. LH stimulates Leydig cells to produce testosterone. Testosterone negative feedback on hypothalamus and pituitary and inhibin negative feedback on FSH together regulate this axis. And final question of this series, question number 10, innovative scenario question is this. A researcher administers high doses of exogenous external testosterone to a healthy male volunteer for 6 months. Which of the following outcomes is most likely to be observed? Option A. Increased spermatogenesis due to elevated testosterone. Option B. Suppressed FSH and LH, reduced endogenous testosterone, impaired spermatogenesis, paradoxically infertility. Option C. Increased LH secretion to compensate for exogenous testosterone. Option D. Hypertrophy of Sertoli cells and increased Inhibin secretion. What is the answer? Answer B. Suppressed FSH and LH, reduced endogenous testosterone, impaired spermatogenesis, paradoxically infertility. So this is the answer. Why and how? Let's see. This is the basis of anabolic steroid induced infertility. Exogenous testosterone exerts strong negative feedback on the hypothalamus and pituitary, suppressing GnRH, FSH and LH. Without FSH, it's needed by Sertoli cells, and LH needed to stimulate Leydig cells for endogenous testosterone. Spermatogenesis is severely impaired, a paradoxical outcome of taking testosterone. So this is exact explanation for question number 10. Out of 10 questions, how many questions did you guess correctly? Please comment below. Today I would like to conclude by explaining these 10 questions. In next series, I will come up with the female reproductive system into the chapter of human reproduction, chapter number 2, biology 12th NCERT. Thanks to everyone. Good luck to all.